Imagine a vast cosmic library. Instead of books, it holds the blueprints of life. This is the world of DNA. DNA sequencing is like reading these blueprints. It's about understanding the order of letters in the DNA alphabet. This order tells the story of life itself. We can uncover the secrets of genes. We can learn how organisms function. DNA sequencing is a powerful tool. It helps us understand diseases, evolution, and more. In this cosmic journey, we will explore two pioneering DNA sequencing methods. We will delve into the world of molecular markers. These markers help us navigate the vast genetic landscape. The Maxam-Gilbert method was a groundbreaking development. It allowed scientists to read the DNA sequence directly. This method uses chemical reactions to break DNA at specific bases. First, the DNA is labeled at one end. Then it is divided into 4 samples. Each sample is treated with a different chemical. These chemicals break the DNA at specific bases: adenine, guanine, cytosine, or thymine. The resulting DNA fragments are then separated. They are separated by size using a technique called gel electrophoresis. The pattern of fragments on the gel reveals the DNA sequence. The Sanger method, also known as chain termination sequencing, revolutionized DNA sequencing. It is a more efficient and widely used method. This method relies on a clever trick using modified DNA building blocks. These modified blocks, called dideoxynucleotides, halt DNA synthesis. They are fluorescently labeled. Each DDNTP corresponds to a specific base: adenine, thymine, cytosine, or guanine. During DNA replication in the presence of DDNTPs, chains terminate at different points. This creates fragments of varying lengths. Each fragment ends with a fluorescently labeled DDNTP. The fragments are then separated by size. A detector reads the fluorescent labels. This generates a DNA sequence. Section 4. Molecular markers, identifying genetic variations. Imagine looking at a vast starry night. Molecular markers are like signposts in the genome. They help us navigate and understand the genetic makeup of organisms. RAPD, RFLP, and AFLP are types of molecular markers. They detect variations in DNA sequences. These variations can be used to distinguish individuals, populations, or even species. RAPD, or Random Amplified Polymorphic DNA, uses short random DNA primers to amplify DNA segments. Variations in these segments appear as different banding patterns. RFLP, or Restriction Fragment Length Polymorphism, uses enzymes called restriction endonucleases. These enzymes cut DNA at specific sequences. Variations in these sequences result in different fragment lengths. AFLP, or Amplified Fragment Length Polymorphism, combines aspects of RFLP and PCR. It amplifies DNA fragments that have been cut with restriction enzymes. Molecular markers have revolutionized fields like genetics and breeding. They help us understand genetic diversity. They also help us map genes and identify disease-causing mutations. Section 5. Conclusion. From sequencing to markers, unraveling the genetic tapestry. From the chemical dance of Maxam-Gilbert to the elegant simplicity of Sanger sequencing, we have journeyed through the world of DNA. We have seen how these methods unlock the secrets of the genetic code. Molecular markers, like celestial beacons, guide us through the vast universe of the genome. They reveal hidden variations and illuminate the intricate tapestry of life. As we continue to explore the cosmos of DNA, we gain a deeper understanding of ourselves and the world around us. The journey of discovery is far from over. It is a journey filled with wonder, excitement, and endless possibilities.